Molecular mechanism of mutation. Any sudden change occurring in hereditary material is called as mutation. They may be harmful, beneficial, or neutral. In multicellular organisms, two broad categories of mutations: somatic mutations and germline mutations. Somatic mutations arise in the somatic cells, passed on to other cells through the process of mitosis. Effect of these mutations depends on the type of the cell in which they occur and the developmental stage of the organism. If occurs early in development, larger the clone of the mutated cells. Germline mutations, they occur in the cells that produce gametes. Passed on the future generations in multicellular organisms, the term mutation is generally used for germline mutations. Some facts. Term mutation was given by DeVries in 1901 while studying evening primaries Onathera lamarckiana. Most of these were chromosomal variations, some were point variations. Originally, the term mutation was given to both chromosomal as well as point mutations. Recently, chromosomal mutations are studied separately. The term mutation is now given only to point mutations. Definition: DNA is a highly stable molecule that replicates with amazing accuracy. Some errors of replication do occur. A mutation is defined as an inherited change in genetic information. Types of gene mutation. Number of ways to classify gene mutations. On the basis of the molecular nature of the defect, on the nature of the phenotypic effect amino acid sequence of the protein is altered or not, on the basis of the causative agent of the mutation. Mutation on the basis of the molecular nature of the defect. Base substitution, insertions and deletions. Base substitution, simplest type of gene mutation, involves the alteration of a single nucleotide in a DNA. A base substitution usually leads to base pair substitution. You can see in this graph or figure. Base substitution is of two types: transition, purine is replaced with purine, and another one is pyrimidine is replaced with the pyrimidine. Transversions, a purine is replaced by pyrimidine or a pyrimidine is replaced by purine. The number of possible transversions is twice the number of possible transitions, but transitions are usually more frequent in vivo. In vivo means within the cell. Insertions and deletions. Second major class of gene mutation, addition or the removal respectively of one or major more nucleotide pair, usually changes the reading frame, altering all amino acids encoded by codons following the mutation, also called as frame shift mutations. Additions or deletions in the multiples of three nucleotides will lead to addition or deletion of one or more amino acids. These mutations are called in-frame insertions and deletions respectively. Mutation on the basis of the phenotypic effects of mutation. Most common phenomenon in natural populations of the organism is called as wild-type phenotype. The effect of mutation is considered with reference to wild-type phenotype. Forward mutation, a mutation that alerts the wild-type phenotype. Reverse mutation, a mutation that changes the mutant phenotype back into the wild type. Missense mutation, a base is substituted that alters the codon in the mRNA resulting in a different amino acid in the protein product. Nonsense mutation, changes a sense codon into a nonsense codon. Nonsense mutation early in the mRNA sequences produce a greatly shortened and usually non-functional protein. Silent mutation alters a codon, but due to degeneracy of the codon, same amino acid is specified. Neutral mutation, mutation that alters the amino acid sequence of the protein but does not change its function as replaced amino acid is chemically similar or the affected AA has little influence on protein function. Loss of function mutations. Complete or partial loss of the normal function structure of protein is so altered that it no longer works correctly. Mutation can occur in regulatory region that affects transcription, translation, or splicing of the protein, frequently recessive. Gain of functions mutation produces an entirely new trait, causes a trait to appear in appropriate tissues or at inappropriate times in development, frequently dominant. Conditional mutations expressed only under certain conditions. Lethal mutations cause the death of the organism. Suppressor mutation suppresses the effect of other mutation, occurs at a site different from the site of original mutation. Organism with a suppressor mutation is a double mutant but exhibits the phenotype of an unmuted wild type, different from reverse mutation in which mutated site is reverted back into the wild type sequences. On the basis of causative agent of mutation, spontaneous mutations that result from natural changes in DNA, induced results from changes caused by environmental chemicals and radiations. Any environmental agent that increases the rate of mutation above the spontaneous is called a mutagen, such as chemicals or radiations. Chemical mutagens. First discovery of a chemical mutagen was made by Charlotte Warbach. Base analogs. Chemicals with structure similar to that of any of the four standard bases of DNA. DNA polymerases can't distinguish these analogs. They may be incorporated into newly synthesized DNA molecules. 5-bromouracil, an analog of thymine, as you can see the figure. 2-aminopurine. Base analog of adenine normally pairs with thymine may mispair with cytosine, causes a transition mutation. As you can see this in this graphic. Both base analogs produce transition mutations. Mutations by base analogs can be reversed by treatment with the same analog or different analog. Alkylating agents, chemicals that donate alkyl groups. Example: ethyl methane sulfonate (EMS). It adds an ethyl group to guanine and produces 6-ethyl guanine, which pairs with thymine and leads to CG-Ta transitions. Also adds an ethyl group to thymine to produce 4-ethyl thymine, which then pairs with guanine, leading to a TACG transition. Mutations produced by EMS can be reversed by additional treatment with EMS. Mustard gas is another alkylating agent, as you can see this picture. Nitrous acid causes deamination, like cytosine become uracil. As you see this graph, adenine changes into hypoxanthine, which then pairs with cytosine. The overall graph in your view. Guanine changes into xanthine, which pairs with cytosine. Xanthine can also pair with thymine. Nitrous acid produces exclusively transition mutations, as we look in this previous picture. So both CG means cytosine guanine transfers into thymine and adenine, and thymine adenine converts into cytosine guanine. Transitions are produced. Thus, mutations can be reversed with the nitrous of hydroxylamine, specific base modifying mutation which adds a hydroxyl group to cytosine, producing hydroxylamine. Cytosine which pairs with adenine instead of guanine. This leads to CGTA transitions, acts only on cytosine, thus cannot revert the mutation produced. Cytosine changes into hydroxylamine cytosine which pairs with adenine instead of guanine, as you see. as you can see this picture. Oxidative reactions. Reactive forms of oxygen like superoxide radicals, hydrogen peroxide, and hydroxyl radicals produced in the course of normally anaerobic metabolism or by radiation, ozone peroxide, and certain drugs cause damage to DNA or induce mutations by chemical changes. Oxidation converts guanine into 8-oxy 7-8 dihydrodeoxyguanine, which mispairs with adenine, leading to GCTA transversion. Intercalating agents: provaline, acridine, orange, ethidium bromide, and dioxin. They are about the same size as nucleotide. They produce mutations by sandwiching themselves, intercalating between adjacent bases in DNA. They distort the three-dimensional structure of the helix and cause single nucleotide insertions and deletions in replication. These insertions and deletions frequently produce frameshift mutations. Radiations, ionizing radiations. In 1927, Hermann Muller demonstrated that mutations could be induced by X-rays. X-rays, gamma rays, and cosmic rays are all capable of penetrating tissues and damaging DNA. They remove electrons from the atoms that they encounter, changing stable molecules into free radicals and reactive ions, which then alter the structures of bases and break phosphodiester bonds in DNA. Ionizing radiation also frequently results in double strand breaks in DNA. Mutation rates: the frequency with which a gene changes from the wild type to a mutant is referred to as the mutation rate, expressed as the number of mutations per biological unit, mutations per cell division, per gamete per round of replication. Mutation rate for achondroplasia, hereditary dwarfism, is about 4 mutations per 1 lakh gamete. Mutation frequency: incidence of a specific type of mutations within a group of individual organisms. Example for achondroplasia, the mutation frequency in United States is about 2 into 10 to the power minus 4. Thank you very much for joining with me. Assalamu alaikum.